as Uma said, I'm Ansil. So I'm a researcher at National Center for Biological Sciences. For my PhD, I studied small mammals, especially rodents, and their associated microbes and their ecology. So for this session, I thought probably bring some of the aspects from the study, but not as a direct results from the study, but also how you study pathogens in ecological community. Something would be basic and will be interest for you. But now, given all of you introduce yourself, like you guys already have some experience with disease ecology or epidemiology or modeling, things like that. So some of these things will be very basic for you. So please bear with me. But also, this session is not a talk, but a discussion session. So I want to hear from you more than me keep talking all the time. So feel free to stop in between and talk and voice your opinions. And that would be very interesting to for the session. What do you say? Ready? Cool. Yeah, so we can start with this question. How do we study pathogens in ecological communities? Yeah. How do we study? So this session is going to be a discussion, and we're going to discuss some of the approaches we can take to understand pathogens or microbes. Yeah, we can do definitely surveillance. Yeah, good. Arjun, what do you want to say? Okay, great, yeah. So I'm still getting familiar to your names, so I don't know your name. So if I know, I'll definitely call out your names and to hear your opinions. So maybe in a couple of days, probably I'll be more familiar with your names. Yeah, cool. Okay, so the answer to this is disease ecology. It's a all-in-one toolbox. Like as you know what is ecology? It's like where we study animals and their interactions between animals and to the environment. But here, what we mostly interested in host pathogen interactions within the context of environment and evolution. So you must have heard about this fungus called chytrid, which actually Yashwan studied that fungus a lot. It's a fungus detected in 1990s, and it causes huge mortality in amphibians in general. So why I brought this fungus here is one of the classical and well-known examples of disease ecology studies. So disease ecologists have found that temperature in and around where the amphibians live could impact how the fungus grows and how it impacts amphibians. So as I said, it causes mass decline in population of many frogs. It made many frogs extinct. So this is one of the important systems people actually started understanding the relationship with pathogens and temperature. But it'll be nice to hear from Yashwan what he has to add about chytrid fungus. Yeah.
Can you hear me? Okay.
Yeah.
So what interesting...
To them.
Okay. So what interesting we find out till now, so there was no study in India before. So 4 or 5 studies was there. So they just did the prevalence thing and what is the presence is there or not. So when in our lab they started the work, so what they found, there's a prevalence was very low. Whatever old marker diagnostic they were using, so based on that they got 4% to 4 to 10% prevalence was there. But there is no symptomatic symptoms were there in the on-field, and there is no die-offs in India. Nothing was there. So they thought maybe it's because of low prevalence. But based on the genetic assessment. So what we found is a mutations in the whatever diagnostic tool we are using, qPCR we are using for detection, TechMan qPCR. So there is a mutations in the binding site of the our probe region. So because of that, so many false negative we are getting in samples. So then we approach a new diagnostic, we develop new marker for that, for detection. So what interesting for what we found, like prevalence was 4 to 60%, 60% it increased 60%. So if total 60% was have positive, but interesting point is that the intensity of the load, means load of the pathogen is very low. I mean intensity is very low. So what we are thinking, there's maybe two aspects. One, maybe our frogs are resistant to that pathogen, and other thing will be the pathogen might be low virulence. So for find out the both things, we need a pathogen. So we tried out the culturing thing also in the lab, whatever protocols are followed in the outside. One in that we are not succeeded there yet to culture the pathogen. What I found, it's maybe because of low load. So because outside it's not like this pathogen is not culturable. It's already cultured. Their infection studies are there. What we are not getting, it's enough load to culture. Because when we culture, that pathogen is very slow growing. So when we culture in the media, so it take 20 days, 20 to 25 days, and its temperature is also it's low temperature growing pathogen. So we can't use antifungal against that. So other fungus will grow faster than in that culture because the load is very low and all this thing. I don't take too much time if you.
No problem. It's fine. So it's a discussion, so it's fine.
Till now, those findings we found till now, and we are doing this thing, capture mark recapture studies also in a field site in Maharashtra to see the disease dynamics along with the population dynamics. Like Uma told that we need to understand community also, so we are doing that thing also. And anything we'll discuss.
Great. Okay. Yeah. Thanks, Yashwant. Yeah. So Yashwant mentioned two things in this thing. One thing is about prevalence and the load. Yeah. So we'll come to that in a bit. Okay. Okay. So now we know about the chytid fungus. It's one of the model system for the disease ecologists to understand the relationship with pathogens and the environment. And also, same time, it's causing the biodiversity crisis by causing the decline of a lot of amphibian populations. So, as I told you, like disease ecology is a subdiscipline of ecology, and it actually uses tools and draw inspiration from many disciplines of science. For example, epidemiology, veterinary science, genetics and genomics, and microbiology, virology. For example, for instance, we need to actually characterize a microbe, like a virus or a bacteria or a fungus. So we need those people also to understand these things better. Then also often we use GIS, Geographic Information System, to understand the locations of pathogen emergence or how they spread over space, etc. Then of course, for an ecology also, the modeling is a very, very integral tool. So that same like that in disease ecology also we use all these things. And of course, public health, and now maybe I can put it in a different words, One Health, like you kind of look at the health of your ecosystem, like animal, human, and plants, all these components under one umbrella. So we can name it, we can draw inspirations, or you can use tools from n number of scientific disciplines to address or answer different kind of questions in disease ecology. So what you see here is basically a word cloud created by it didn't create, but that I actually asked the ChatGPT to actually give me 100 words which related to disease ecology. So it spit out all these words. Then I made a word cloud out of this thing. You can see that there are surveillance, as you said in the beginning, then food, demography, migration, host parasite, GIS, control, microbiology, all pathogen, transmission, dynamics, prevention, SAR, all these things are coming in this thing. So the point is that there are a lot of things you can use, a lot of scientific disciplines you can use to address different questions in disease ecology. So before we go more into the details, so I think it's better we just discuss some of the basic terminologies. Yeah, that would be interesting to look at. For example, we mentioned that here parasites and pathogens, right? So the whole disease ecology would be like a study of parasite and pathogens and their interaction with the host. So there are different kind of classifications people do. One kind of classification is divide the parasites into microparasites and macroparasites. As the name suggests, microparasites, you can see them like with your naked eye. Sorry, you can't see them with your naked eye. You need a microscope or something to see a bacteria or virus and protozoa and fungi. But also remember, some of the protozoan parasites are actually big enough you might be able to see also. Then you have macroparasites, which you can see with your naked eye. They are like helminths, like a worms, parasitic worms. Then arthropods, like ticks, fleas, and often mosquitoes. Yeah, all those things. But we use this term parasites and pathogen interchangeably very often in disease ecology. Even I use it like a parasite. Oh, this is a parasite or a pathogen. So, but when it comes to pathogen, it's a very specific thing. So can you define a pathogen? I showed like, what I've written there, but I'm just going back. Yeah? What do you say? What could be a pathogen? How do you define a pathogen? Do not be strictly defined in textbook forms, but I'm just saying, what is a pathogen? Exactly. So pathogen is a microbe. It can be any of these things. It causes the damage or causes the disease in an organism, or it has a potential to cause the disease in organisms. So here you see this word called susceptible host. So do you know what a susceptible host is? Exactly. So that pathogen capable of infecting that host is basically it's a literal word, susceptible. This organism is susceptible to that pathogen. So let's look at what is a host. Here is susceptible host, exactly. So it's an organism housing a microbe and allow it to live and replicate. So that we can broadly define as a host. So in the context of Hendra virus, so you heard about Hendra virus? So Hendra is actually a river in Australia, close to Queensland. So this virus detected in Australia. So they named after that place. So this Hendra virus is it's a paramyxovirus. It's an RNA virus hosted by bats. Bats are known reservoir for this Hendra virus. So the virus can spill over from bats to horses and from horses to humans. It's directly also is possible, but the established route of transmission is from horses to human. So in the context of this pathogen, Hendra virus, the horses and the humans, they are hosts, right? So then when it comes to reservoir, okay, fine. I should have asked you before I showing that. So what is a reservoir? What do you think? What could be a reservoir? Without damaging the host.
It can replicate there, but it's not damaged.
Okay.
Stores a pathogen.
Sorry?
Stores a pathogen.
Organism stores a pathogen. Okay.
Compulsory for the life cycle of the pathogen.
It's compulsory for the life cycle of the pathogen? Not compulsory?
Like vector need a life cycle completely.
Okay. Okay. And you? Okay, it's a carrier with no symptoms, right? Same thing. Okay. Okay. Yeah, I think all these answers are correct. But what they define is a population of species community in which microorganisms naturally occur and indefinitely maintain. So It's basically you find this pathogen all the time in this species or a population or community. As you said, often it's true that these pathogens are not causing any symptoms in the reservoir host, but that's not necessarily true all the time because we don't know what are the immune responses these hosts or reservoirs do have against these pathogens. So, but casually we speak, we can say that we don't see any clinically manifestation of these pathogens in these reservoir organisms. But what all you said is correct. So now, here, this white-footed mouse, so Uma actually mentioned this during, well, she was mentioning Lyme diseases. So this white-footed mouse is a reservoir for a bacteria called the Borrelia burgdorferi. It's a vector-borne pathogen. So when these vectors, like ticks, bite these mice, they get these pathogens. They can actually transmit that to humans, not just humans, other mammals in the community. So white-footed mouse is a reservoir. Yeah. So then the other thing comes is vector. So vector is basically an arthropod which carry a pathogens from a reservoir to an incidental host or a susceptible host we talk, or it can also carry pathogens from one host to the other. But like when I mentioned the case of Hendra virus, it's actually often humans get the virus from horses to humans, right? From bats to horses, horses to humans. So horses comes in between this transmission pathway. So let me be clear, horses or any mammals who acting as an intermediate host are not vectors. Yeah, vectors are mostly arthropods. Yeah. So here in the case of Lyme disease, I just mentioned the white-footed mice as the reservoir. So these black-leg ticks are the vectors for this Borrelia burgdorferi, that pathogen, a bacterial pathogen causing Lyme disease. Okay, so then there's another term we always hear associate with the spillover. We'll come to spillover in a bit, but shedding. The shedding is basically the process of dissipating the pathogen into the environment. For example, if these bats do carry pathogen, that's not true, always bats carry pathogen. So if they have it, so some pathogens can come through aerosols, some pathogens can come through fecal samples when they defecate, or something can come through urine. So that pathogens are coming to the environment through this process is called the shedding. Yeah, so that is an opportunity for any other organism or any other susceptible host to contract that pathogen and transmit from that point. Yeah. So then, again, we're coming to the spillover. So for example, let's say these bats are, or any other organisms that matter, shed viruses to the environment. So it can, other organisms can get contracted. So here we have a host as an example. We call the host as a spillover host, and the bats are reservoir host. So the transmission can be directly or indirectly. So this spillover host can get the pathogen. Pathogen can establish in that susceptible host and replicate and further get transmitted. So this process is called spillover. So here the host is a naive or a novel host, never been exposed to this specific pathogen, but the pathogens are jumping from bats to horses. So here you can actually replace a human also. You can get the pathogens from the bats too. So when you do that, that becomes the zoonosis. So a spillover event between a reservoir host and the human, we call it zoonosis. Yeah, it's not necessarily have to be the first novel incident, but it can also repeatedly happen. So here you have these rodents, like you can see this red thing is a bacteria or a pathogen. And then the transmission can be direct or indirect. Here you have these ectoparasites be transmitting this pathogen between the reservoir and the human. Yeah? But you see, there are not all the individuals of the rodents do have the pathogen. So that is what Yashwant was mentioning, something about prevalence, like how common a pathogen is in a population. So we'll discuss that a little more in a bit. Yeah, so given we mentioned spillover, one important thing we need to understand is spillover is not that common as you think. The moment you encounter a rodent or a primate or a bat or a bird or something, or you handle them, not necessarily you're going to contract the pathogens it carry. Because even if it's a reservoir, the reservoir organism might not be carrying the pathogen in high intensity all the time. So to have a successful spillover event, so we have actually sessions, detailed sessions on spillover in the coming days. You'll hear more about that. But to briefly mention this, there are a lot of barriers for a pathogens to cross before it causes a successful infection in the target or spillover host we mentioned. The host was the targeted, target spillover host I mentioned, right? So there are multiple barriers. So these barriers are actually attribute of reservoir, pathogen, environment, and the host itself. So let's quickly look at how these things are playing out here. So for example, the first two barriers, you can see what it is, reservoir host distribution. Where the reservoir found, it's important for that pathogens to get contracted to the host, incidental host, a susceptible host, something. Yeah, so then reservoir host density. Let's say a very common, abundant, high density reservoir versus a rare or like a low density reservoir. What would be contribute more risk of transmission? High density, of course, right? High density, we are likely to encounter high density host than the rare species. So then comes to the pathogen prevalence, what Yashwant mentioned. So if, let's say, you have 100 rodents, a reservoir for pathogen X, but 90% of the rodents do carry this pathogen. But now let's say a bat, which is again 100 individuals, but only 10% of the population carries the pathogen. So you're likely to encounter the pathogens from the rodents because they are highly prevalent, right? Versus the bats, which is rare, like they're abundant, but the prevalence of the pathogen is very low. So then comes the infection intensity. So the infection intensity is the load, what Yashwant spoke about, how abundant the pathogen is within an individual. Let's say a bacteria or a virus have for me, I'm carrying like a 1 million copy versus 10 copy. So that's what it means by infection intensity. Then what matters is like a pathogen released from reservoir host and how it gets spread in the environment. For example, sometimes it comes through the fecal matter of the reservoir host, or sometimes you need a vector like a mosquito or a tick or something to carry this pathogen around from host to host or reservoir to host. So that also matters. Then when humans get exposed to these things, not necessarily we will be susceptible. Like there are multiple mechanisms which actually make us resistant to infections. One is like we directly contacting the pathogen in our hand, right? So some pathogens need a cut or a blood barrier crossing to enter into our body and cause infection. So as long as that is not there, so that is like a dead end for the pathogens to spill over, right? So then again, when it comes to our immune responses, we have active immune system which actually detect foreign body and clear off something. So that also kind of leads to an unsuccessful spillover event. But anyway, the point is that you can see these barriers do have certain holes, right? So all those things has to be aligned well to have a successful spillover, right? So it's kind of make the spillover event is actually a rare event. It's not something like we think. It's the moment you touch a wildlife, it's something. So the moral of the story is that it's okay to touch a wildlife or go near a wildlife. You don't have to be scared. Cool. So anyway, and one more important thing, all these barriers we spoke about vary in space and time. So it's kind of an additional layer of complexity adding to this system and make it extremely, extremely rare. Yeah? Okay. Now, let's move on. Before we move on, the one thing is, so I kind of picked up those terms, reservoir, host, vector, spillover, all these things from this paper is actually came out in 2021 during the pandemic time. So that time there was a lot of miscommunication and a lot of random usage of these terms. So they kind of very nicely explain what a reservoir is, at what settings we can use this thing, and what is a host, what is spillover, things like that. I really encourage you to actually go through this paper, and it would also help you kind of catch the terminologies in the coming days. But many of you might be already familiar with, but still I encourage you to just have a look at it in the coming days.
What we might probably do is, of course, all these lectures are being recorded, but we'll also make a Google Drive link or something and a folder where we put all the presentations and also papers. Sometimes there are so many papers, you don't know what to read. Or maybe you're like me, you download them and don't read them. It's good to have that more secure.
Yeah, that'll be a cool idea, Ma. We'll do that actually. We'll put together whatever the paper we are discussing or like I uses for these things. I'll put it in a Google Drive and we'll share with all of you. Yeah, that'll be, and you also feel free to share anything you think it's very interesting and for us to, yeah. Okay, now we'll come to the crux of the our session is how do we study pathogens in communities, especially ecological communities or wildlife? Yeah. Of course, humans also we can study. We do touch upon that. Yeah. How do we study? Let's forget about the questions. We have it over there. I asked this question in the beginning also. You said we'll do surveillance, we'll do field work. Yeah, that's all true. Yeah. But fundamentally, these are the like a two, or I can break down into three questions people ask, like how pathogen impacts species. For example, chytrid fungus, they impact amphibians. And also, you heard about this pathogen called white nose syndrome fungus. It's a fungus which infects bats, and it actually causes decline in bat population. And so, how pathogen impacts species is one of the important questions people do ask. For example, malaria, how malaria impact survival of birds. For example, in Hawaii, like Uma mentioned actually, Hawaii, a lot of birds actually died off or they showed population declines once the malaria got introduced. Then the other question is, what pathogens are maintained in animal communities, or how they are maintained? These are the two important questions disease ecologists generally try to answer by studying pathogens in ecological communities. So one of the most primary thing we do as disease ecologists is like we do field studies, like we do go out in the field and collect samples and you test them in the laboratory and kind of understand what's going on, what's out there, what's going on there, what are the trends and patterns we can see. Yeah, so I won't go details about how do we do field work or how do study animal communities, because we do have dedicated sessions coming on those. So the one way of doing is you can do cross-sectional study, like a one particular time point, you go to a place of your interest and you sample multiple populations and you kind of look at or detect pathogens and they look at their distributions, something like that. But of course, before that, you need to have a question and a hypothesis ready to explore, right? And also, often people do exploratory studies kind of just to understand, especially when you're placed there is no baseline data, you need to do exploratory studies to understand what is the microbial diversity, what is the animal communities or host communities, etc. So one approach is to do is cross-sectional studies by looking at one time point, multiple populations, and find out what pathogens or microbes. So I will be interchangeably using this microbe and pathogen, but you be clear, all microbes are not pathogens. Pathogens are only ones can cause infections or damages to the host, right? Okay. So for example, we did some study, some cross-sectional study. We kind of took these two sites. One is called Cardamone, and the other one is Kudremukh. The Cardamone is actually a forest plantation mosaic. You have forest, grasslands, and tea plantations and human habitation next to each other, kind of a mosaic. Then you have a protected area in Kudremukh. Yeah, so you might have heard about this name, Kudremukh National Park and all. So we sampled different species of rodents there. So Rattus satare, then 3 species of mus, and then we looked at this prevalence of this microbe called Bartonella. So the what Bartonella we are talking here might not be pathogenic, but many known species of Bartonella are associated with the human infections. So I'll be use this Bartonella, and we looked at how common they are in different species. So you can see that the most common in the species called Rattus satare over here, then followed by this Mus fernandoni or this mus over here. Then they're quite rare in other 2 mice. Okay, so this is an example of how we do cross-sectional studies. So then the other way of you can do field work or field surveys are do setting things in longitudinally, like you sample something continuously for a longer period of time. Let's say every month you go and collect these rodents, like the rodents I worked on, you go and collect the blood samples from these rodents and test for Bartonella every month, right? So then over a period of time, I collect this data and kind of, I can understand how the prevalence of this Bartonella changes over time. So, but there are many other tools are there, not just by catching the host, like a rodent or a bird or a bat. You can do certain other things like a wastewater sampling. Dr. Farah will talk more about wastewater sampling in the coming days. Then you can sample soil, and some of you are doing air, and you can also, we do something called under the roof sampling. It's kind of, I'll talk about that in a bit. So anyway, you can collect these environmental samples and kind of try to identify what microbes are there, what are pathogenic, and how their presence and distribution changes over time. Yeah. So here, some of the, so this is some image from UNEP program. They collecting the wastewater for the wastewater surveillance. So colleagues from TIGS also do a lot of wastewater sampling, and it's one of the like a biggest study which is continuing for more than 3 years now. 2 years now. Yeah. So then we also do some studies like we go and find these bat roosts. So Darshan is here. Darshan is the one leading that study. So I think this is Darshan. Oh, one of this is Darshan. So yeah, so we kind of go to these roosts and spread a plastic sheet. So we don't catch any bats, but you have to wear these safety dress, as we call a PPE. Yeah, so we collect these bat poop and urine, guano, and then we bring back to the lab and test and find out what kind of viruses or bacteria is there in this thing. And specifically, Darshan is interested in coronaviruses. Darshan, you want to add a little bit about your study? Because I'm the only one talking.
Like Ansil mentioned, initially we started off with cross-sectional studies to understand what pathogens are present, not like prevalence, because initially we didn't know what pathogens are present. So we took up unbiased approaches to understand what viruses are present in bat communities, and then we narrowed upon what viruses are important to focus on. Basically, then I was more interested in coronaviruses because these are more epidemiologically important. We know that they can infect many species and all, but what uh, lineages or like species of coronaviruses we found were more of found only in bats, not in other mammals. So we are using this system to understand ecology of these viruses. So based on life history stages of the bats, there could be varied shedding of the virus. Suppose during mating season in bats, so many individuals will come together, and these assemblages can cause increased contact between the individuals, and there could be more transmission of pathogens, and there could be a spike in shedding of these viruses, as well as if there is many juvenile bats. So they might not have immunity developed. They might lose the maternal antibodies they receive from their mothers, and there could be again a spike of viruses they have. So we are trying to understand these things as of now.
Thanks, Darshan. Yeah. So that's what Darshan is doing. It's a kind of longitudinal study to understand a prevalence of a specific microbe or pathogen, we don't know, how it changes over time. So this is a very interesting tool to kind of understand temporal variations in pathogen prevalence. For example, here you can see the study. It kind of shows the prevalence of a pathogen here. It's a coronavirus, basically, and how it changes over time. It's 2 years, you can see. So initially there's a peak, then it comes down, then it increases again. So the period it increases is when there are more juvenile bats in the colony. So it kind of tells what explains this seasonal variations you see in the system. And so overlaying other variables into this variation what we see is very important. For example, this study is actually kind of shows the positivity of Hendra virus over time, and they bin them separately when there is food shortage and when there is no food shortage. So the emerging studies are actually shows that when the bats are stressed, when I say stress, it can be reproductive stress, or it can be resource limitation, like there is a scarcity of food or something. That's what you see here. So when there is stress, or here the food shortage is there, the positivity is actually increasing. So my point here is that, so these kind of longitudinal studies are extremely useful to understand underlying processes and what causes the seasonality or variation in the pathogen prevalence we see. But also, sometimes we need to take some precautionary measures when we do this under-the-roof sampling. So here, what you see, the 3 different ways, or like 2 different ways the sampling is being conducted. So I told you, when we go to under the roost, we spread a plastic sheet and divide that plastic sheet into 4 quadrants, and we collect samples from that. So let's look at this first example. So it kind of shows the actual true prevalence of this pathogen in these host species. So that is basically 1/6, that's 0.17. That's the true positivity or prevalence of the thing. But when we do the quadrant wise, we are collecting all the samples from 1 quadrant and considering as 1. Okay, so you might end up getting a 1/8, 2/8, that's a 25%. Then the another approach is you put the plastic sheet, I said Darshan is doing. So you collect everything, right? So then what you get is a 1 positive out of 2 samples, right? That's kind of overestimated prevalence It's 50% here, right? So we really need to be careful and how you sample these rows. And so here is the best is to do probably like a grid-based sampling. So you take, so that's what Darshan is doing. Kind of, you will be close to what the actual prevalence is. But again, it's something impossible to kind of match the exact true prevalence. But what you can do, minimize the difference between the two. So here actually it's represented very well. You can see again the prevalence of Hendra virus over time, and the green is the pooled sheet, this scenario, and you see it's almost 75%, right? Then comes to the pooled quadrant, which actually like a between 25 and 50. Then individual, while individual is very close to 10% or something like that. So, but you see certain bumps here also. It's kind of a spiking, but what you're capturing is not the actual prevalence in the community. So, but people also did some simulations kind of to reduce the minimize of this bias. Kind of like if you know a bat colony with 100 bats, how many sheets you need to put out to actually minimize the difference between the actual prevalence and your sampling methodology. So these kind of things you need to be careful about when you do under the roof sampling. I thought it would be good to mention here as we talk about under the roof sampling or field surveys. Okay, so now we collect samples in different way, and now we need to understand what pathogens are there or what microbes are there in the samples we collected. So the one approach to look at is like, especially when we don't have any idea about the microbe community or viral community or a bacterial community, what do we do is like something Darshan just touched upon is called unbiased approach. It's a hi. So what I meant was you actually catch the individual. You catch the individual, take a poop sample or a blood sample and kind of test them. So that is different from what you get from the fecal samples using any of these approach. Does it? No, we're sampling as much as possible. Yeah, not one bat, yeah. So then if it's only catching one, if we get a positive individual, that's like 100%. And if you're not getting anything, it's like a 0, right? It'll be multiple individuals, yeah. This three sampling is just a hypothetical example of what kind of pitfalls we can expect from these sampling technique. But our study is actually in one area. Yeah. So, especially if the species you're working with is a protected species, you might not have always access to catch that individual and collect the pathogen, right? And also it will be stressful to, sorry?
Yeah, but I think he's also asking about pooling.
No.
Just about
why are you doing both?
Non-invasive versus invasive?
How it makes difference when you're sampling with the individual plants and you are collecting the fecal sample. If you're doing that in the same area, same region area, right, does it matter difference? Because as the graph shows, the individual, if you're collecting from individual things, it is very less prevalence. If you are collecting the fecal matter, it is much more higher.
One thing is pooled, and it has an ecological significance. So one thing, you might not be able to sample your host species or reservoir species all the time, right? You can't catch them. Second, what we left with one of these approaches, right? So then we can minimize the gap between these individual level thing and the pool thing. Given you know how many individuals are there, what is the prevalence you are expecting, things like that. Okay, so then why we need to understand the pathogen presence in the environment is that that's what kind of contribute to the spillover or something we talk about. Right, the presence of pathogen in the environment. For example, I'm walking into a bat cave, or I'm going to somewhere the bats are, or under the roost or something. So how much pathogens are there is what matters. That's what I'm going to expose, or any host in that matter going to expose. So quantifying this also important. Yeah. Correct? Yeah. Sorry, each quadrant will be having? So it's a quadrant. So, but there are methods which uses grids within the quadrant. For example, let's say this is the quadrant, this is the sheet. Darshan is spreading under a roost. Okay? So, and we divide that into 4 quadrants. And here there are like 3 samples, 3 urine or 3 fecal. Here there are like 5. Here there are 3. Here there are 2. So all these samples are actually goes as 1. Yeah. So might have 1 positive, might not have 1 positive. Yeah, coming from. It depends on how many individuals in the colony we study do have viruses or pathogen in that matter, or any microbe. So, but there are also approaches like you subdivided this into sub-quadrant and you take them. It kind of maximizes your sample size, and probably you might get closer estimations. Sorry? Definitely, it's better than pooling everything in the sheet versus having these quadrants. Yeah. But also, it matters like how many sheets you spread for how many bats. Yeah, I show you. So that's a good question. It's a problem when you have a bats with multiple species, like a multi-bat host colonies. You will have multiple species coming into. So it's a challenging situation. So what we do, especially in one particular time where Darshan samples, there's one slightly bigger species comes and roosts with the smaller species. So the poo pellets of these bats are slightly bigger. So we don't collect them, but also it's quite possible accidentally we can make these mistakes. So once we have a pool, Darshan does this, Darshan do species ID, and kind of make sure what predominantly found in that sample is this one. Yeah. But it's kind of biased. Yeah. Yeah.
This is why the prevalence data on its own may not be so useful unless you have other covariates to understand the drivers of this. That's why he's talking about the seasonality and size of the roots and other factors.
Yeah. Yeah, absolutely. So in such situations, even we can actually use presence of another species as a variable and see suddenly, let's say, this another species coming into the roost, how the prevalence changes. So we can use all these variables to explain the patterns we find. Yeah.
So when you talk about a particular, you're working in a particular area,
Yeah.
so are you looking at multiple root sites in that particular area, or just one root site? And would you find variation in multiple root site in similar area?
It's quite possible, given, so at the moment we are only looking at one roost, yeah, of one species, and then we have another site with multiple species are roosted. But predominantly we are focusing on one species, and once in a while another species coming into the roost, kind of one roost. But I would expect the patterns of prevalence to vary in a roost from another place. Yeah, so I would expect a spatial variation in prevalence thing. Assuming the environment they experience might be different. Yeah, it would be nice to have something really synchronizes with between roosts. Yeah.
Yeah, so one more.
Yeah, go.
So if in one area you just have one species and another there is an accumulation of 3-4 species, and there's same pathogen in both areas, but one area you might find it to be a very specific for it to be very specific, but in the other, given that there are multiple species, the pathogen has now turned into a generalist. Will that be something?
It's a tricky question. I'll also think a little bit. So you're saying in one side we have only one species, and the another side we have multiple species. Here the pathogen is specialist, yeah, and here it's not. It's everyone carrying it.
Yeah.
Now the question is, the species you find in site 1, is it there in this site 2?
Yeah, but given that this accumulation is quite old, so the pathogen has now turned into a generalist here because it's better for its survival, maybe,
Yeah.
or spread.
Why can't we think like this pathogen always was a generalist, but here
It turned into a specialist, yeah, both ways.
no, but it's, I think it's the pathogen is generalist, because here where you study the site 1, there's only one host is there.
Yeah.
So we don't have other hosts to compare and say that, okay, this is a specialist. I would say, I would call this pathogen a specialist only when you have a species and the other species you find they're also here, but they're not infected, something like that. You get the difference,
Yeah,
right?
yeah.
But in the other case, there is a community, the assemblage, it's multi-species, or the roost has multiple species, and they are having these pathogens, same pathogens, right?
Yeah.
So holistically and looking at the whole picture, I would say that pathogen is opportunistic and generalist. It can infect multiple hosts at a time. Yeah.
Okay.
Right? Yeah. Yeah.
Give an example, of a curiosity, where the pool sheet is more preferable than, the pool sheet is more preferable than the pool quadrant, or is that not the case?
From this one, we have not done any such experiments, but based on this thing, I would say if the objective is to kind of find out what truly the prevalence or something matched to the true prevalence, I would say quadrant is always, that's what it says, quadrant is better than the pooling the whole thing. It's kind of...
Never another scenario where research question where the sheet would be more preferable than...
Sheet would be more preferable than quadrant. I can't think of anything, but would help if we have all these data at the same time and kind of correlates so that's exactly what these people did, and it says it's not working. Yeah, it's kind of overestimating kind of prevalence. Yeah, but yeah, I can't think of anything. What do you think? Anything? I don't know. Anyone?
It's also a logistical issue, right? Capturing facts every time may not be very plausible.
Yeah.
But there could be some questions where this difference may not be that much of an issue.
Yeah. Especially, like this difference is not that an issue, especially you are interested in what is the load in the environment, things like that. Yeah. Is that what you're trying to say? Yeah. Yeah.
The studies just like exploratory, like you want to do a cross-sectional study to understand exactly prevalence.
We just want to know what disease, like what pathogens are there.
Yeah.
And then do studies.
Yeah. So I would actually put this paper in the drive and share it with you guys. It's really an interesting paper. They kind of correlated between how individual prevalence and pooled and pool chord rate and number of sheets varies. Yeah, so it would be worth a reading. Yeah. Yeah. At the moment, I can't think of anything like a pooling everything into one would be a better strategy versus having chord rate, unless what we are interested in just to understand the pathogen load in the environment. For example, you can do these techniques called RT-PCR. You all know, we do all our COVID tests and all we must have done, no? So you can quantify number of copies of viruses in the samples. So I think that kind of given a proxy for an idea about how many or how much pathogen load is in the sample or in the given area that matter. Farah?
Sampling sites are much more in number, then I think pool sheet will be much more easier to deal with. Suppose 100 sites you are collecting, then you cannot go for pooled quadrant. So you can just check in which site there is a prevalence, and then you can go for pooled quadrant or individual level. So it would be much more easier to,
Yeah, it depends on your question, exactly, right? What you want to do with the sample and also the logistics.
Where we could use this pooled sheet instead of pooled quadrant. So that's where this
yeah, you're right. Yeah. So I think if we only want to know like what it's in the samples, you can probably use the pool approach. Yeah, versus the prevalence. If what you're interested in prevalence or something, chordate would be better. But if what you're interested in is only about the diversity present in the fecal samples or the urine samples, probably you can use a chordate approach. Thanks for that. Yeah. Okay, we'll move on. Cool. We collected samples now, like Darshan said, we wanted to find out like what it's there in your sample. So one way is to kind of do an unbiased sequencing. This is a technique we call metagenomics. You can sequence DNA or RNA or any nucleic acid in that matter from your sample and kind of use bioinformatics to find out, or you can use your reference databases to find out what is present in your samples. For example, when you start your study, often you might not have the information about what is the diversity of microbe you're dealing with, or what is the pathogen you are interested in, or what are the pathogens present in that matter. So it's kind of a starting point, especially when we have no prior idea about the microbial community you're handling with. So for example, here this study recently came out, is actually talks about viruses, different viruses, RNA and DNA viruses. You can see the viral family in this left side from these geckos and skinks. And here in the bottom, you have a different samples and it's colored, collected from different locations. And here what you see is the color is the relative abundance of different viral families. You can see calciviridae, adenoviridae, and which I also don't know. Then herpes viridae, hantaviride. So basically showing the presence of different viruses and its relative abundance from these samples collected from these lizards. And the important thing is, so you can, one thing you can look at the presence and the abundance of these viruses in the samples, and also at the same time you can actually extract the sequence data and use that for further analysis. For one such analysis, something here you can see from about the hantavirus, they actually took the hantavirus sequences and kind of build a phylogenetic tree. It kind of shows the relationship between other hantaviruses and the viruses they harbor. Okay, other hantaviruses from other species. So kind of like a evolutionary relationship between this hantaviruses and the known hantaviruses. One thing. The other thing, especially in these things, you will get large amount of data. So often you get the whole genome of the virus, for example, hantavirus. So, and then what you can do is you can use bioinformatics to, one thing is kind of, when we talk about these viruses, what we are interested to know whether they are pathogenic or not. One is like use the bioinformatics tool to kind of predict the pathogenicity. Even these days, like machine learnings and artificial intelligence is actually used to understand whether, based on the sequences, can they be pathogenic or not. You can actually find out what receptor in the virus or what motif in the virus is actually can bind to the receptor in the host, something like that. So it's kind of, once you have the sequence data, you can also predict whether they are pathogenic or not, or we can say, like Uma said, whether they have the potential to cause infection in other host, something like that. Okay, so the other approach is like a microbiome analysis. So like here we are not really sequencing the whole genome of the microbe you are interested or the virus you are interested. Here there's also called 16S amplicon sequencing. So the 16S rRNA gene, it's a universal gene, and there are primers, the primers which bind to this gene, and you can, using this polymerase chain reaction, you can amplify that region. So it works for all the bacteria present in your sample. So you can amplify that, then you can sequence all of them, and again you can use bioinformatics pipeline and reference databases to identify what they are. Again, all these things depends upon your reference database. If your reference database is good enough, you get good resolution up to species level. Often you might not have that luxury, so you end up having phyla level or family level or genus level, something like that. So, hi, yeah. No, yeah, Uma. Everybody speak with the mic, okay? Raise your hand and I'll get the mic to you. It's not on, Uma, yeah. They have to press this thing. Yep.
What you're saying is right, that approaches where we work with non-invasive samples like fecal samples, you will need some amplification process. And also, I'm divining what I think you're saying, that if you don't know what you're looking for, then it becomes problematic. So usually for bacteria, for example, we use 16S, we use ITS for fungi. Viruses are a problem. Like, what do we use for viruses, right? And so, for example, there are other approaches like very expensive whole RNA sequencing, which is what Darshan, for example, does. But I think you wanted to add something?
The mic, fine.
For viruses, we usually go for polymerases. So they are highly conserved across the genuses and stuff.
Yeah, but what she's mentioning is actually not the targeted thing. So she's asking, like, you have a metagenome approach, but what if your DNA is very less? Is that what you're asking? So there are two things. One thing is, if I understand your question correctly, you need to you need to throw out a lot of your background from your sample. For example, in our lab, what we collect the scats, and often what we need is the host, or let's say pathogen. So what we need to throw out here is the host DNA. So there are these enrichment methods, you can use it to separate your pathogen component and your host component, and you can use that one.
Yeah. That's a good point that you brought up. Enrichment is something which, in fact, what we are doing, we are using it actually to get more of the host. But you can do it was developed for the opposite, to actually get more of the bacteria based on methylation patterns, which differ between, birds, mammals, and vertebrates and eukaryotes.
Yeah. So then the other thing, these metagenomics tool or sequencing, whether let's say you do RNA-seq or any of those things, some of the methods do also have this, for example, this method called rRNA depletion thing. So it's kind of a filtering step where you throw out all the host RNAs. So then what you left with, I can say in another way, the proportion of your microbes you are interested in is higher than the host. So such tools are there to kind of maximize your output of microbial DNA in your sample which is poor.
Also, you can employ this capture probe-based sequencing. So
That
it's
again becomes
completely
target,
not
right?
unbiased, but
Yeah, it's not unbiased, right?
Yeah, it's a little bit biased, but
Especially if you know what you're looking at to get the whole genome, you can use these probes.
I don't think the question was.
Her question was about what I answered, no? So what is it? Basically, if you have a very low quality DNA, yeah. Sorry?
Can I reverse this question?
Yeah.
This is for the use industry. You are sampling things, say in an environment, how do you know when you have not found something that your method was correct? We are randomly sampling that you can do metagenomics just because you haven't found a certain sequence. Doesn't mean it was not right. How do you actually test that your sampling was correct, that it was definitely
First thing, like we said, we don't know what are there in the samples. Either we can go for a targeted one like 16S, we can just amplify and see whether bacteria is there. ITS we can go for whether fungi is there. So there will we get an idea. The sampling method was correct. It's getting amplified. So the microbes, whether it is bacteria or pathogens are there. So the issue we were facing with that amplification in a metagenomic approach, we want to understand what are all the pathogens present, not pathogens, the microbe present in that whole environment. So if we go for 16S, we will only getting the bacterias. So that was the question I was asking. The mixed population, even the nucleic acid, the DNA is very low also. Do we have any method to enrich the microbes so we can do metagenomics?
Yeah, so I think that, Farah, what you bring up is really important. And I think in general, that is something which hopefully tomorrow we'll get some insight on. What I said initially, right? To prove absence is very difficult. It's not possible. And that is why statistical approaches and sampling design are critically important in these studies. So hopefully, we'll get some more insights onto that tomorrow.
Yeah. Yes.
So I had a question. You mentioned the reference material, the reference data set is required. So where do you get this reference data set?
So there are a lot of reference platform. For example, NCBI hosts a reference platform. So you can use already existing data set in all these repositories, like EBI metagenomics do have a reference database. NCBI has a GenBank basically has a reference database. Those are the reference I was mentioning.
Okay,
yeah.
Thank you.
Actually, when I was first time I'm hearing about the reference thing, I was thinking, okay, you also have to create something to compare with. That's what you must be confused with, right? Yeah, good, very fast. Yeah, okay, so now I think we have only half an hour or something left. We just rush through some of these things and kind of wrap it up. That doesn't mean that don't ask questions or don't interrupt or something, okay? Yeah, so the other way we can do is do these microbe studies, like microbiome analysis. So predominantly, we use these things for understanding gut microbes and all. But now people also moving to understand microbiome associated with the different sample types. For example, this study, one study we did, and kind of comparing the microbiome of the species I mentioned before, Rattus satara. It's an endemic rodent from the Western Ghats. So how the microbiome associated with the organ samples, like we are lucky to get some individual sacrifice and we got organs. So like a liver, spleen, kidney and all those things. And then you have the swabs, like oral swabs and rectal swabs of each individual collected. And you kind of comparing how the community looks. And what you find here is a different community in organ tissues versus swab samples. And also we kind of overlaid the pathogen I mentioned, Bartonella. If there is any impact of Bartonella infection causes the difference in microbiome or something, which we don't see anything here, then we can also take one more step and kind of find out what these bacteria are. That's where the reference library, different database coming in. So the sequence read we obtain, the 16S, you can actually compare, or it's a process of blasting, kind of, but you do through a bioinformatics pipeline, and kind of find out what phyla are there. You can see there are differences between these two sample types. For example, in swab, you have more of Firmicutes and proteobacteria. When it comes to tissues, you have more of Bacteroidetes and less of Firmicutes, things like that. Then again, you can take one more step and kind of find out what exactly they are at genus level. You can see different genus of genera of bacteria we detected in 2 different samples, and how their relative abundance change between the things. So these kind of approaches you can take, and it's also kind of give you an information about what bacteria are there in these samples, which probably I want to study. For example, when I started my PhD, I didn't have an idea what pathogens to target. So kind of collecting some fecal samples from these rodents and doing a 16S microbiome analysis kind of gave me a profile of pathogen communities. For actually that led for me to actually choose Bartonella, which is common in tissue samples, to take it as a one target pathogen for my PhD. Yeah. So then again, if you already know what pathogens you want to work, and then you can use these targeted methods like a PCR or RT-PCR. In RT-PCR, you basically, or qPCR, you can kind of quantify the pathogen. But here you get to amplify, and then you can visualize the amplification with a positive or negative or something. So then you can sequence them. Then once you have the sequence, you can use the sequence for one thing, again, to build phylogenies, and you can look at diversity within the pathogen. And also you can use it for understanding the relationship between pathogens and the host. For example, in the studies, basically a plant and pollinator relationship is showing, but you can think of a pathogen and the host here. So these lines actually shows that the interactions between different pathogens and different hosts. Yeah. So then there are also other approaches like serological methods. You directly don't target the pathogen, but also you look at the previous exposure of your pathogens in your serum samples. So you collect blood and you isolate serum from those organisms, and you test them using these different techniques. For example, ELISA, we do a lot of medical tests using this ELISA. And the Luminex assay, and also some, these days people do something called the serum proteomics. You have a serum sample, you kind of look at all the proteins present in the, it's like a 16S microbiome approach of a serum sample. Yeah, you sequence all, or like you do a mass spectroscopy and you get different proteins, and you can, if it's a known protein, you can kind of associate what is overexpressed, what is underexpressed, what is the function, and things like that. So here, actually, there's one study from the lab kind of looked at filovirus reactive antibodies. Like so what you see in this x-axis are different group of filoviruses. Like this is Ebola virus, Bundibugyo, Typhorus virus, rest in Sudan, all those viruses. And then y-axis, you have the fluorescence came out from these reactions. So these dotted lines and the solid lines are the cutoff drawn by different approaches. And what comes above those things are the positive samples. So essentially, these two species, Eonyctris phalia and Rusetus leschnolti, these two are co-roosting fruit bats. They do have a previous exposure to one of these pathogens, or pathogen something similar. It's not confirmatively saying that they are Ebola virus, exposed to Ebola virus or filoviruses, but something similar to that, or something showing similar reactivity to this viruses. Then, this is not a molecular technique, but still I just wanted to mention this because conventionally, or traditionally, we use a lot of microscopy to understand the presence of pathogens, especially when we study blood-borne pathogens like malaria, trypanosoma, etc. We actually make thin blood smears. I think if you go to hospitals, we still do make smears, right? So you make thin smears and you use different staining techniques and kind of visualize them under the microscope. So you need to be a little trained to understand what pathogen looks how, at what life stage of the pathogen looks what, something like that. So it's been traditionally, we use this a lot. So then also you can understand vector ecology. First you can do vector surveillance, especially you can see how vector populations changes over time. We'll hear more about vector population changing in a couple of days' time. So here what they've shown is like Aedes mosquitoes and Culex mosquitoes and their population, how it changes over time. And what important is that if we have these kind of trend, you can overlay many variables on top of it. One thing is directly look at how the case burden changes over time. The second is like you can overlay environmental variable like rainfall, temperature, all those things kind of understand what are the underlying drivers of these population fluctuations of vectors. Then the other thing is you can do vector behavioral studies. So when I say vector behavioral studies, like how the vectors respond to habitat changes or change in environmental variables, or what are the life cycle and what life cycle causes infection, something like that. For here, what you can actually see, a study which Uma mentioned, this is from the Keesing's group. So here, what you see in the x-axis are the fragment size, small to large, and what you have in the y-axis is the density of infected black-leg ticks when I say infection is the Lyme disease one, you remember, right? The Borrelia burgdorferi I mentioned before. So what you see here, the relationship is that as the fragment become bigger and bigger, the density of infected ticks are actually reducing, right? So why is happening? So the one way this can happen is that the one Uma mentioned is the rodentification. So when you have smaller fragments, the first group of animals are going extinct or locally missing is large herbivores. And when you have a large herbivores gone from your fragment, what happened? More room for rodents to grow, right? So one thing, rodents are granivores most often, and there are more productivity of the grass, so they have more resources. Then also some studies actually shows that large herbivores do pack soils. So once they have been taken, so there are more loose soil for the rodents to make burrows. So basically they are reproducing more in the absence of herbivores in smaller fragments, their densities goes high. As a result, the tick density or the infected tick density also goes high. This could be one way this is happening here. But anyway, by studying these vectors also we can understand what's happening with the pathogens and pathogen transmission. Then comes to host pathogen interaction studies. Like we can, if you have we have all in our institutions do have a laboratory mice, right? So we can do control experiments on such animal models to understand how infection progresses and how immunity develops or how pathogens evolve over time. So for example, this study is actually on canaries. They kind of injected malaria in one group, and you have a control group with just blood from a non-infected bird. And then they actually tracked the microbiome on the top, and the same time they also tracked the infection. So they kind of found out like the microbiome is different from the infected and non-infected birds. And also they found out like after a few days, the infection goes away in these birds. So my point here is that these kind of control experiments you can do kind of understand how pathogens or evolve or how immunity develop and things like that. So here is another study from Egypt and Romania. It's a native range of house sparrow. So they looked at the immune response by looking at the response of these birds against this compound called the phytohemagglutinin. It's a plant compound. When you inject, it will have a response against this thing. So what they found is, in Romania population over here, the response was much higher in both the male and female versus the Egypt. So there are studies which actually shows that there is variation in immune response in population, but what interesting to know is what driving these variations, which not discussed in the study. But again, we can do such studies to understand the interactions within, like in vivo interactions of pathogens and the host. Then, of course, as I mentioned in the beginning, we use lots and lots of models in ecology. Same like that in disease ecology also, we use models. So the models essentially kind of help you understand the underlying pattern in your data through this process called the model fitting. So it's kind of you, let's look at here, you have years here, and the y-axis you have a number of disease outbreaks. You kind of approaches the system with a question and a different hypothesis, and you try to fit this line over your data by optimizing different parameters and land on a best fit model. And that explains your data. For example, here, you remember I showed you this before, the coronavirus prevalence, how it varies over time. So this peak here and the dip over here is actually explained by the period where the bats are breeding or they give the birth. So that's what causes the infection. So once you have these kind of trends, you can actually extrapolate and predict these trends for the future. And that would be very efficient or very useful for kind of planning, mitigation measures and things like that. But one thing I'll say you, or not all models are accurate and also not well explain your data. So we will have a dedicated sessions coming, and in the coming days also we have a different models will be discussed. So, but there are two kinds of models generally we use in disease ecology. One is statistical models. They are like a pattern-driven model, kind of understand the pattern in your data and the relationship with other variables. So for example, generally we use these models like general linear models, like linear mixed models and additive models. They're all extension of linear regression and kind of help you understand your data better and explain your hypothesis. For example, we did a study about the Bartonella. We were curious to know what drive or what correlates with, what variable correlates with Bartonella prevalence in the host. So we kind of first thought, okay, probably this is the host density is causing it, probably it'll be the habitat itself, or it'll be site level variation, all these things. We kind of thrown in all these variables in the thing, and what came out, or what we realized, that the ectoparasites load in a species are the one actually causing the Bartonella thing. I won't say, I should turn that word causing, because what often we find out is a correlation. So it's not a causation. So it's a kind of, we can say, this ectoparasite load is correlated with the Bartonella prevalence in hosts in different habitats. Then the other thing, some people, this study is actually did the generalist linear mixed model to understand what are the variables explaining immunity in vampire bats. So the vampire bats generally feed on mammal blood, so they do like livestock also. So you can see all the variables they tested, and you have this beta coefficient here. The higher the coefficient, the stronger the relationship. That's what simply you can put it. So you can see that livestock and reproductive stage, reproducing individuals actually have a higher immunity. So I kind of bind them together and kind of say that higher livestock kind of supporting healthy individuals, and they are reproducing more, and they also happen to have a high immunity, something like that. So we can use these models and kind of understand your data better. Then you have a, yes?
What is the negative value of the beta coefficient?
It's probably a negative relationship. Yeah, it's not a strong positive relationship like that. Yeah. So then you have a process-driven models, like for example, what we're investigating, like why this is happening, what this is happening through models. So let's say suddenly the prevalence of malaria increasing, due to deforestation. So we can test such processes using these mechanistic models. So one of the famous example is SIR model. Different varieties of SIR models are being used in disease ecology and epidemiology. So here the SIR, S stand for susceptible, I stand for infected, and R for recovery. So the population have all three groups. For example, when the infection starts, you have all the individuals are susceptible. As the infection or the time progresses, they comes down, the infected increases, and after some point, the number of infected people in the populations comes down, but the recovery increases. So we can use these approaches to understand how pathogens are transmitted within the populations. Yeah. So anyway, it's time to stop. So before I stop, I actually, yeah, 1 o'clock? Yeah, I think we'll stop for 1.5 hours. Question session? Time stop for me, yeah. Yeah, so we have half an hour for questions. Yeah, so I thought I'll just stop with this. So if you remember, the whole session was about different approaches we use to study pathogens in ecological communities. But I have not covered all the approaches. For example, I have mentioned anything about GI, how we use GIS to study pathogens, or how we use microbiology, virology, veterinary science. All those things I have not mentioned. But fortunately, we have experts from these backgrounds coming in the coming days, and we'll be discussing those things in detail. But before I stop, I wanted to take you through, I want to tell you like how these fields are heading at the moment. So now the people are thinking about interdisciplinary approaches, like integrating field work, laboratory experiment. For example, I remember that controlled experiments we did on the cannery. Yeah, and also kind of do mathematical modeling. You bring all these things together towards understanding one problem or one system better. For example, here they are interested in, it's a kind of hypothetical framework came out recently as a perspective. So it's kind of, they want to study bats and their viruses. So you can use immunological and other targeted approaches to understand pathogens present in these samples. Then you can identify what are the stress, like ecological variables we collect, and we can bring them back to the habitat. But at the same time, you can also do mathematical modeling, understanding the system, and also you can do experiments in laboratory settings. For example, let's say stress causes higher viral shedding in bats. So the one way to study is that so you might see the pattern, your data might show that in the field, but you wanted to actually also demonstrate that using like in laboratory experiment to rule out it's not a correlation, it's a causation. So what you can do is what people are proposing to do, you can actually create cell lines from these hosts you are interested in, and you can mimic the stressors, what we are calling here, like a presence of stress. So there'll be stress hormones or something. You can add the stress hormone to your thing, and also same time you can add viruses and see how the viruses are responding in presence of stress. So then you can kind of understand what gets overexpressed or what gets underexpressed, and what's the relationship association between these pathogens and the host. So then that information you generate from this thing, you can feed back into your model, and then again you can take back to your natural world and probably plan intervention, things like that. So it's kind of overview of where the field is heading globally. But again, we have so much work to do in India. So again, I'll stop here, and we can have questions, of course, and we can discuss more. Yeah. Yes. Pass the darshan. It's got it. You have to Yeah.
Yeah, hi. Thanks for the talk. My question is regarding the serological methods that you mentioned. I was just wondering that you may not always get antibodies and antigens, right? So then, is it even important? But at the same time, I was also thinking that if you don't see antigens and antibodies, that means they are not causing any issue to the animal or any disease or any symptom. So maybe that's enough. I mean, is it making sense? What I'm saying?
Yeah, I don't have a right answer to that, to be honest. You are very good chance sometimes you miss all these things. For example, one thing, the titer of the antibody, if it's not in a high titer, you might not pick up one thing, unless your assay, what you're looking is extremely sensitive and specific. But the other thing, what if the immunity or the antibody wanes off, like, after some point, you lose the immunity. For example, in the case of COVID, when the new variant comes, we are not all immune for all variants, something like that. That also could cause
Yeah,
things.
actually, my question is more of, so let's just say there are many microbes, but they are not really causing anything. That's why you don't have any antigen antibodies.
It's true.
So then what's the point of doing, I'm assuming it's more expensive, like molecular?
I think serology are cheaper than molecular things.
Yeah, so then serology is enough, right? Because then it will just tell you whether it's, so when do you incorporate molecular with serology? When is?
So one thing is serology will only, most of the time serology captures the previous exposure. So you might not be an active carrier of that pathogen or a microbe at this point of time. So we can only infer that at some point in the past you got exposed to this thing. So other than that, we don't have any other data to talk more about it.
Okay.
Yeah, we don't have a sequence data or something like that. Only presence, absence, or also the one thing you can get it, how abundant was your antibody response or the fluorescence, something what we talk about here. So that could be a proxy for, I don't know, I don't know how to say that. Yeah, but it could be for the exposure.
Okay, thanks.
Yeah. Yes.
But I think that's a really good point. That these are all proxies, right? Like you sequence some DNA, you sequence some RNA, but like we said, we have no idea what it's doing. Serology could have been exposure in the past, could be recently, we don't know how long that exposure was, but it does mean immunological response. So in some sense, it's maybe more meaningful than just sequencing some DNA or RNA. So ideally, it would be nice, but that's why I think maybe that disease ecology is a field which has really there's not been a lot of disease ecology in India because it's so interdisciplinary, requires so many different approaches, and in general in India everything is very siloed. We don't work well together, so it's important. Sorry, was that a negative or bad thing to say?
No. Maybe that's where we start.
Yeah, so I think that it's really nice to try to look at all of these things in similar data sets or something like that. And just one small example with some of Ansil's samples, we had a master student called Aslaha. She actually looked for toxo, both with a serological test, which is actually for humans, right? That test?
It was designed for humans.
Designed for humans.
It could work.
So she actually tested rodent sera with the human toxo serology test. And then she also did a DNA-based amplification for toxo. And she very luckily, there was very low positivity, which we were surprised at. We thought toxo would be very high prevalence. But for the same sample, she got positive DNA as well as serology positive. So it first of all shows us that some approach from humans we are using is working with rodents, which is really cool now. We can actually survey more widely. So serology, by the way, is much cheaper, right? So serology can be probably done widely, and then we can use some kind of hierarchical approach, like start with serology, then quickly hone in on something else, some kind of something like that.
Thanks, Uma.
Hi, thank you. So you're talking about that chytrid fungus which affects the frogs. It was dead in, I can see the image, it was overall dead in that river. It doesn't affect the fishes in the river. It's only specific to the amphibians.
Yeah.
Okay, so I have a very basic question. It might be silly.
We welcome basic questions always.
So, like, it's very difficult to understand this reservoir and amplifying host. These are always having a contact with interactions more with humans. So, why this pathogens is having very special criteria to choose a specific model organism as amplifying host or reservoir? Is that any like, for example, I can say the Culex mosquito is a vector for Japanese encephalitis. Its amplifying host is pigs, and host is humans. The humans who are all rearing that pigs will be getting the Japanese encephalitis, and the reservoir is egrets. So it's all related with ecological conditions. Like egrets will be seen in the rivers or lakes, and the pigs will always be in the contact, and from there it's coming to the humans. So is there all its connection, or it has some criteria for the pathogens to choose a specific model organism?
Okay. One angle I'm thinking, but I would like to hear many voices in this question. It's a really good question. Things about compatibility thing. So when a pathogen tries to knock to the system of a host, it's or the entry into the cell and replication, immune response, all receptor mediated. So if a pathogen has a docking point which is not complementary to a receptor in a host, it's not going to be successful there. So that's what would be my take on this thing. But I would like to hear what Guha thinks or Farah thinks about it. Yeah. Yeah. That won't work, no? Ashwin, just pass. Yeah, this one. Pass.
I can do it. Yeah. Two things. So I think I agree with Ansil, but I want to start with one basic observation. We tend to be, my name is Guha. I'm a participant here. I'm from Korea University. So I mainly work in disease ecology, in disease dynamics, in natural populations. So we can meet more formally a little later. But I just want to mention one thing. So we tend to be very loose with our language, and we have to be a little careful about it. So when you ask a question that the pathogen chooses, it almost looks as if, it's like, oh, am I going to choose vanilla ice cream or chocolate ice cream, right? This is all, and I'm as guilty of this as everybody else is, but I just wanted to mention that when we mention things like the pathogen chooses or anything of that sort, we mean it from a purely from an evolutionary times, right? So basically, what we are loosely saying is that the pathogen has evolved over time to be able to infect or not infect particular groups, right? So I think that I just want to mention that one thing. But I think on the whole, I agree with what Ansil says, that basically what happens is that these are all kind of random mutations. It depends upon what kind of mutations the host cells have, what kind of mutations the pathogen has. When these two fortuitously work out, the pathogen is able to infect the host, otherwise it's not, right? For example, SARS-CoV-2, very good example of this. We still don't know. So we know that the original host was the horseshoe bat. We don't yet know what that intermediate host is that allowed it to jump into humans. But we know that there was some intermediate kind of stopping point. But the fact of the matter is that SARS-CoV-2 had certain mutations that allowed it to kind of infect humans, right? So I think that that's basically what happens. Sorry, go ahead.
Yeah. The mic. Okay, here, press that on.
Sir said exactly that a random mutation. So as she said, whether the pathogen choose the real host to amplify. So pathogen is not in the perspective of evolution. It is not choosing. It is rational and random. So it is a chance. It is a chance whether that host support for amplification or just to support for its growth. Acts as a susceptible host or acts as a reservoir, it all depends upon the host, how it interacts with the pathogen. So here the pathogen won't select or choose the host, it is random. Again, soon after the selection, it decides whether that host is a susceptible host or a reservoir or a vector. So it all depends upon the random. It is, evolution is not directed, it is random. So it is all depends upon, say as a scientist said that it's a mutation. Mutation is also a kind of criteria for the pathogen to ensure its survival in nature. Rather, choosing one pathogen, though it is successful in one host, it's no guarantee that, say for example, as we use medicine. So if it will die, if we design a drug against a pathogen, it will die. So meantime, the pathogen also struggle to evolve, even in presence of the drug. So in a different way. So that means it puts a pressure on the pathogen to survive in all dimensions. As again, you can find out the mutation which supports for its survival even in presence of the drug. So it all depends upon the final conclusion of the evolution of life is to existence and survival. So these two words, which explains the coexistence or the pathogen or the vector or the reservoir. This is my information.
Yeah.
Thank you.
I just want to say, I think what largely we are discussing is mostly the context of viral pathogen. Like, see, she specifically mentioned JE, which is also a viral, and I think for a lot of viruses as well. But if you really think from the parasitic organisms like malaria, lots of mosquito species bite so many hosts, right? But not every mosquito species can transmit that parasite. It's a compatibility issue. Same for humans as well. There's so many different types of malarial parasites, birds, mammals, and also rodents, everything. I cannot believe that we never get bitten by a mosquito which is carrying a bird malaria, but we never respond to them, and same for the birds, So it's all very specific. It depends on the system, of course, and how specific and compatibility it will. But yeah, viruses rule the world because they can mutate and they easily, transform or exploit other hosts.
Thanks, Farah.
Okay, so since a lot of them are like random mutations, the models which are used right now, do they also have a stochastic component into the models, or is it still working on? Do we have a model in the SI models which have a stochastic component in it? Is like, yeah, the evolution of the pathogen since it's random.
So actually, okay, let me step back. So initially, when modeling started, right, the major kind of limitation was computational. These kind of deterministic models, which were purely mathematical based, were very computationally easy, right? And so I think that's why, for the most part, those kind of models kind of took the front seat in terms of modeling these kinds. So, for example, the SIR model, it's a deterministic model, which has actually given us a lot of really cool insights into how diseases work. When we are talking about stochastic models, increasingly we are finding that more and more studies are focusing on stochastic models, and the only reason we are able to do that is because now there are no computational limitations per se. Even a very ordinary like a PC or something is capable of doing pretty much most of these kind of stochastic models. But yes, stochastic models are becoming more and more common now.
Yes. With respect to the modeling for the virus, I mean, this is something which has been a very complex thing for, say, SARS-CoV-2. Nobody knows how the virus is going to evolve. This is why people rely on the RO, how many individuals it can infect, and that is a very good estimate. When you know the RO for a pathogen, like for measles, it's really high, it's like 14 or something, which means that it can infect. And same for every variant of SARS-CoV-2 has had a very different kind of RO. Once you know that, it becomes a bit easier at a population level to model it. But from your mutational point of view, it's really, really hard to know that in large scheme of things.
Does everybody know what RO is? R naught? Yeah. So R naught is what is called the basic reproductive number, and so in terms of pathogens, it's basically the number of new infections that one infected individual can lead to. There's an important caveat, in a predominantly susceptible population. So obviously as Ansil pointed out, if you just think about this simple SIR model, you start off with pretty much everyone being susceptible, then most of them get infected. Then we have this kind of recovered or kind of refractory kind of a population. If most of the recovered individuals again become susceptible, then this is not a major criteria, but usually what happens is that there's some level of immunity. So the RO is calculated on that like initial thing of when most of the population is kind of susceptible.
Thanks, Ram.
I just asked, when we're mentioning these terms, stochasticity and determinism, could you briefly explain what they are?
What is stochasticity? Okay. So as we get more and more basic, the answers actually get more and more difficult, right? So I'll say that much. So what we mean by stochasticity, at least from a modeling perspective, is this kind of random variation, right? But remember, it is random variation within some limits. So for example, you may put some kind of a distribution on that random variation, right? It was, for example, when Ansil showed you that regression line, right? What is a regression line? A regression line is basically a best fit line, right? That goes through these points, right? What you're trying to minimize in this regression is your mean square error. That is like how far each point is from that regression line, right? Now, what is the error that you put? Basically it's a normal distribution of error around that line, right? So just like that, every kind of regression. So for example, a binomial regression will not put a normal error, it'll put a binomial error, right? So basically that is what we're saying in terms of this kind of stochasticity, right? So basically what I want to mention is that it is not completely random, right? It is random within some kind of biological reality, is what I wanted to mention. Yeah.
So, yeah, I mean, can I also add?
Yeah.
So, for example, in the SIR model, you have susceptible and infected and recovered population. If you want to introduce, say, stochasticity in the model, that, I mean, say, is a vaccination drive going on. We don't know if any new variant might appear. So when people are modeling, they kind of look for different scenarios. What could, because it's not just 3 parameters, right? So you have to think of different things which might be affecting lockdown, stuff like that, which could affect the thing. And when as many things they can account for, depending on the disease you are trying to model, I think it becomes model a bit better or robust as I would.
I think so. Like you said, can you add evolutionary aspect to this models? For example, if you consider the link from S to I, you parameterize all these equations using some time scale parameters, how fast they go from one compartment to the other. In short term, that might remain constant for a while, but if you want to look at for a longer time scale, this beta might be a function of time. So that's how this RO will change with different waves, right? The thing is, if you increase more complexity by incorporating so many things, it might be difficult to get data on time to calibrate the model for that setting. So usually, like for short term, simplistic models would, might be good enough, but people can go really wrong also. For COVID, we have seen a lot of absurd predictions.
I would definitely agree with this. So simpler the model, the better. So the more parsimonious model, the better. There's no question about that. So yeah, and there was one more thing I wanted to mention with what you had said. Now I'm blanking. It'll come back.
Great. Yeah. More questions? Oh no, we're close to lunchtime. Yeah, I think it's. Yeah. Then. Yeah, fine. Then, if you have any questions, we can discuss in the coming days.